The next pandemic will not begin in a nature preserve. It will begin in a text editor. We have officially bridged the air gap between digital code and biological reality. Today, biology is no longer just chemistry, it is software. It is stored in insecure cloud databases, transferred over standard networks, and compiled by automated machines. And like any software, it can be hacked. In the legacy era of biosecurity, defending against a biological threat meant physical containment. You secured the physical viral sample behind steel doors, airlocks, and armed guards. That defense architecture is now obsolete. Modern biotechnology relies on distributed synthesis. A researcher, anywhere in the world, designs a genomic sequence on a laptop, uploads the blueprint file to a cloud database, and transmits it directly to a high-throughput DNA synthesizer. The machine reads the digital file and physically prints the DNA from base chemical canisters. No human hand touches the raw material. No physical sample crosses a border. The pathogen has been digitized. It travels at the speed of light across networks, bypassing physical border controls, customs, and traditional containment protocols entirely. To prevent the accidental or malicious creation of restricted pathogens, the industry relies on screening as a service. Before a machine prints a requested sequence, software checks the digital design against a database of known dangerous organisms. This screening software is the only gatekeeper, and it is highly vulnerable. Security researchers have already demonstrated the concept of DNA obfuscation. By splitting a restricted genetic sequence into fragmented, seemingly harmless data packets, attackers can bypass the automated software filters. The screening system sees nothing but benign, fragmented data. But once those separate, non-restricted sequences arrive at the automated lab, they are physically assembled into the active, lethal agent. The attack vector is not biological replication. It is a data injection vulnerability. The vulnerability does not stop with the sequence files. It extends to the physical hardware. Modern laboratory facilities are fully integrated Internet of Things environments. They rely on automated liquid handling robots, remote-controlled centrifuges, and cloud-managed environmental sensors. Most of these devices were built for scientific collaboration, not national defense. They run on legacy operating systems, lack encrypted firmware, validation, and are frequently connected to corporate networks. If an adversary gains remote execution access to a facility's automated systems, they do not need to steal anything. They can silently alter the purity of a batch of vaccines. They can reprogram systems to mix unintended hazardous chemical precursors. They can turn a legitimate pharmaceutical manufacturing facility into an unintentional bioweapon factory with a single remote configuration change. For the global strategist, this convergence demands a fundamental shift in risk calculation. If you operate in the pharmaceutical, agricultural, or national defense sectors, your biological risk is no longer separate from your network security. A compromised cloud database, a weak credential, or an unpatched IoT vulnerability on a lab floor is now a vector for physical destruction. If your leadership team is still viewing biosecurity through the lens of airlocks, they are defending against a 20th century threat. The digital-to-biological bridge is open, and the perimeter has ceased to exist. Follow and subscribe for more. Trescudo, we track the signal.